A severely drooping flower, drained of its moisture, can rapidly regain its structure and stand upright after being placed in a vase of fresh water. Conversely, raw mangoes submerged in a saltwater brine will shrivel and lose volume as they transform into pickles. This shriveling effect has been used for centuries to preserve food. Packing fresh meat in thick layers of salt prevents rot by removing the moisture required for bacterial growth. These three events, the reviving flower, the shriveling mango, and the preserved meat, all result from the same physical mechanism. These organic materials are constructed from cellular structures bound by continuous, thin sheaths called membranes. These membranes act as gatekeepers for a process called osmosis, which regulates how solvent molecules move into and out of organic matter. While these membranes appear to be solid sheets, they are actually porous at a microscopic scale. This cross-section of a semipermeable membrane shows a network of holes constrained to a highly specific size. Small solvent molecules, such as water, can slip through these pores, but larger solute particles are physically blocked. Because of this selective barrier, solvent molecules will flow from a side with lower solute concentration across the membrane to the side with a higher concentration. Osmosis is the physical drive to equalize this concentration difference using water as the moving agent. Since osmosis is a consistent flow toward high concentration, researchers investigated how to halt this movement before the volumes become completely imbalanced. In this U-tube, solvent crosses the membrane. As it dilutes the concentrated side, the liquid level physically rises. This flow is stopped by applying downward pressure, forcing the liquid levels to remain even. The exact amount of excess pressure required to stop the passage of solvent molecules into the solution is called osmotic pressure. This isn't just a chemical exchange. Osmosis creates a measurable physical force that pushes against the membrane barrier. Because osmotic pressure depends on the number of solute particles present, it provides a method for measuring the mass of microscopic substances. In dilute solutions, the osmotic pressure, P, is directly proportional to the molarity, C, at a specific temperature. By expanding this formula to include known values for volume and weight, we can algebraically isolate and solve for M2, the molar mass of the solute. Other methods for determining mass often require boiling or freezing, but osmotic pressure measurements can be taken at standard room temperature. This makes the method useful for studying proteins and biomolecules that would otherwise break apart or lose their structure when heated. Then, measuring the pressure of water movement, scientists can determine the weight of large, delicate macromolecules without damaging them. This physical process occurs constantly within the human body. Our red blood cells are essentially small volumes of fluid contained within semipermeable membranes. To prevent unwanted water movement, medical saline is calibrated to 0.9% salt. This creates an isotonic state where the pressure inside the cell matches the pressure of the fluid outside. If cells are placed in a hypertonic solution with a salt concentration higher than 0.9%, water flows out of the cell to dilute the exterior, causing the cell to shrivel. In a hypotonic solution, where the exterior salt concentration is low, water flows into the cell. This causes the cell to swell and eventually rupture. These cellular reactions show that maintaining a specific salt concentration in the blood is a physical requirement to prevent cells from collapsing or bursting. In everyday life, high salt intake can cause water to move into tissue cells and the spaces between them, leading to the swelling known as edema. This same force is responsible for moving water from the soil into plant roots and up through the rest of the plant's structure. Even the survival of salted meat depends on this. The salt creates a hypertonic environment that draws water out of bacteria, causing them to shrivel and die. From the preservation of food to the stability of human blood, osmotic pressure acts as the physical regulator of fluid balance across biological membranes.
When a raw mango is placed in a concentrated salt solution, it shrivels. Within a few hours, the internal water leaves the fruit, transforming a firm mango into a wrinkled pickle. We see the opposite effect in a vase of fresh water. A wilted flower placed in the water springs back to life, its stems and petals regaining their rigid, upright structure as they fill with fluid. These same physical changes occur at the cellular level. We have to examine the microscopic architecture shared by all living things to identify the silent force regulating the movement of water. In each of these examples, the substance is bound by a membrane, whether it is of animal or vegetable origin, like a pig's bladder or a parchment. This continuous sheet serves as the primary boundary. While these membranes look like solid films, they contain a network of submicroscopic holes. Because of these pores, they are classified as semipermeable membranes. Small water molecules pass through the membrane's pores, but larger solute molecules bounce off. When placed between pure solvent and solution, water flows through the barrier. This natural flow, called osmosis, continues until equilibrium is reached, moving from lower to higher concentration. The membrane dictates this movement. It is not a passive wall, but a filter that ensures water movement is dictated by the chemical concentrations on either side. Since osmosis is a physical flow of matter, it can be resisted. It is possible to apply enough force to stop this movement entirely. If we apply extra pressure on the solution side, we can halt the flow. The pressure required to just stop the passage of solvent is known as osmotic pressure. This pressure is a colligative property, depending only on the number of solute molecules. For dilute solutions, osmotic pressure, P, is proportional to molarity. Using P equals CRT, we calculate the exact force generated. Because these measurements are taken at room temperature, this method is used to determine the molar mass of proteins, polymers, and other macromolecules. It allows researchers to weigh delicate biomolecules that might be destroyed or become unstable if they were heated. By calculating the precise force needed to stop the movement of water, scientists can identify the building blocks of biological systems without damaging them. In clinical medicine, understanding these pressure levels is a requirement for patient safety. This is most visible in the management of human blood cells. Two solutions with the same osmotic pressure are called isotonic. A 0.9% sodium chloride solution is isotonic to the fluid inside blood cells, which is why it is safe to inject intravenously. In a hypotonic solution, where salt concentration is less than 0.9%, water flows into the cells via osmosis, causing them to swell and potentially burst. Conversely, in a hypertonic solution with over 0.9% salt, water flows out into the surrounding fluid, causing the cells to shrink and collapse. Cellular health is not a fixed state, but a balance maintained against these constant osmotic forces. These microscopic laws explain the phenomena we observe in our daily lives and global industries. We preserve meat with salt and fruits with sugar to create a hypertonic environment. When bacteria land on salted meat, osmosis draws the water out of the bacterial cells, causing them to shrivel and die before they can cause spoilage. This same force enables plant growth. Water moves from the soil into the roots and up into the plant, partly due to osmosis, while a limp carrot becomes firm again as water moves back into its cells. Even human edema is a direct result of this process. When we consume too much salt, our bodies retain water in the intercellular spaces to dilute the concentration, leading to visible swelling. From the movement of water in plant roots to the precise requirements of intravenous medicine, osmosis provides the mechanism for fluid regulation. Whether in a single human cell or across an entire agricultural system, osmosis is the force that maintains the necessary distribution of water in living organisms.